Good morning. My name is Oliver Plümper from Utrecht University in the Netherlands. I'm one of the co-PIs and managers of the Excite network, and I would like to spread out a very warm welcome to everyone to this first Excite network workshop on electron and X-ray microscopy of Earth materials. We hope this workshop could take place in person in Utrecht, but we all know the current situation, so we have to make the best out of it and do this workshop online, but I'm sure we will have two very exciting mornings and afternoons until 3 o'clock with a lot of information around electron and X-ray microscopy, specifically focused at an introductionary level. So this is not an advanced workshop. We will have more advanced workshops within the Excite network at later stages, but this first workshop will give you an introduction to electron and X-ray microscopy. And if Gerd here, if you have not started recording yet, please start the recording. A couple of things beforehand, please mute your microphone and turn off your camera while we're giving the presentations. Please feel free to post questions into the chat, and we will try to do our best to answer all these questions. If they can't be answered after the presentation, we will answer them later in Gather Town or tomorrow during the question and answer session that is specifically targeted at electron and X-ray microscopy questions for you. So there will hopefully also be time for questions after each talk, and we will be recording the presentations. So if you don't want to be in the recording, please turn off your camera, make sure it's turned off. So who are we and what do we provide? What is the Excite network? Very briefly, if you have not been on our webpage yet, please go to www.excite-network.eu to find out more about what we have to offer. Besides these workshops that we're planning and doing now, our main purpose of the Excite network is transnational access to high-end microscopy facilities around Europe. I'm here in the Netherlands, but we have places in Belgium, and France, and Spain, and Italy, Germany, UK, and Norway for now. And the current call for applications to access electron and X-ray microscopes free of charge is open. So I suggest you go to www.excite-network.eu to find out more about how you can access all our microscope facilities. We also do community-driven research, specifically targeting electron and X-ray imaging of Earth materials, and we may say more about this later. And the third point is we do a lot of networking training, and this is our first official training workshop aimed, as I said, on an introductionary level around electron and X-ray microscopy. What is the scientific challenge? Many Earth material or Earth processes, be it devastating earthquakes, natural occurrences, volcanoes, processes within subduction zones or around ore deposits, or be it the storage of hydrogen, geothermal energy, are all controlled by the physical processes happening within minerals and rocks. And we need to try to understand how we can determine the physical and chemical processes operating in Earth materials, and we try to do this through imaging. So we are confronted with a vast amount of scales, from understanding nanometer scale processes all the way to kilometer, and Excite operates at this bridge between nanometers to millimeters at the micro to macro structures of rocks and minerals. So what we would like to bring to the world is a multi-scale, multi-dimensional correlative characterization approach of Earth materials, from 3 dimensions using, for example, X-ray tomography, to 2-dimensional imaging using electron microscopy, again to 3-dimensional imaging using all kinds of nanoscale techniques around electron and X-rays, all the way down to understanding the atomic structures using, for example, transmission electron microscopy. And we will be touching on all of these different parts throughout the next 2 days. So to give you an overview about what we have planned for you within this network is, today we will be focusing on electron microscopy, and tomorrow on Tuesday, the 18th of January, we will be focusing on X-ray microscopy. So today we start off with an introduction to scanning and transmission electron microscopy. And in an online workshop, we can't cover everything, so we will touch on specific parts. Then we go into chemical analysis and crystallographic analysis using electron microscopy, and also crystallographic analysis using transmission electron microscopy, to then in the end more advanced techniques around EM and machine learning approaches to how to characterize microstructures. Then we will have some lunch, and of course in between we have a couple of breaks. Please turn off your microphone if you have not done so, by the way. And then we will gather in GatherTown to have a poster session and a meeting to chat about science, and Gertje Termart will tell you more about GatherTown before we go to lunch, of how to access GatherTown and how you can access the posters there. And then, as I said, tomorrow we will have the day focused on X-ray tomography and X-ray microscopy, going from the basics of X-ray imaging to post-processing to more applied parts of chemical characterization, multi-scale challenges to dynamic imaging, and then after lunch we will have a question and answer session in GatherTown on how can electron and X-ray microscopy solve your research question. So this is where you can bring your question of, hey, I have this idea, or I would like to write a proposal for transnational access. What do I need to do, or what type of microscopy technique could solve or address my research question? And then last but not least, tomorrow afternoon, we have a very exciting keynote lecture by Milay Poshoy on From Dust to Bugs, Transmission Electron Microscopy as a Versatile Tool in Environmental Mineralogy. So I would highly suggest to also come to this keynote lecture, which will give a lot of amazing insights into state-of-the-art electron microscopy. Without further ado, I would like to right away jump into an introduction to scanning and transmission electron microscopy by showing you these three sketches here. Most of you know a light microscope where we have a light source, we have a sample through which the light is going, and then we can observe this with an eye. Very similar to a light microscope, we can use electrons to image materials. And we typically subdivide this into two types of electron microscopes: a scanning electron microscope, where we have an electron source, we have some lenses, as well as in the light microscope, but now these lenses are electromagnetic lenses. Then we bring the electrons onto a specimen, and it's called scanning electron microscopy because we scan the electron beam across the sample. And then we have detectors and a computer that can then change the interaction of the electrons with the matter into information that we can process. And then we have a transmission electron microscope, which is actually very similar to a light microscope, but in this case here turned upside down. We have an electron source now going through the specimen, and we observe with the eye on a fluorescent screen, for example, the information we get from electrons transmitted through a sample. So this transmission electron microscopy was actually the first type of electron microscopy, as it doesn't necessarily require a computer. And then with the emergence of computers, scanning electron microscopy became possible. So I would like to introduce briefly how scanning electron microscopy works and what kind of techniques we have there. And then I go briefly into transmission electron microscopy, and we will pick up these techniques over and over again today. So in scanning electron microscopy, where we scan the electrons across the sample, we typically subdivide into three types of information. We can get topographical information, so looking at the height changes of a material. Here you see little spheres that actually you see how they're arranged with each other, and we get a topographic information. We can get chemical information through imaging using so-called backscattered electrons, which I will explain in a minute, or through using X-rays that are emitted when we bombard the sample with electrons, and there will be a specific lecture on using X-rays and scanning electron microscopy later today. But we can also get crystallographic information by doing diffraction inside a scanning electron microscope. And there will also be a dedicated lecture on how we can use this to get crystallographic information and what type of information we can get and what we can do with it later today. So why use electrons instead of photons? And what are the differences between electron and photons? This mainly comes down to the wavelengths of an electron, where we can determine the wavelengths of an electron seeing this equation right here, and you can subdivide this in a non-relativistic and a relativistic equation. But the bottom line here is that the wavelengths of electrons are much shorter than those of photons, and so we can image much smaller features. If we look at this, here we have the wavelengths of electrons at 10 kilovolts, so the acceleration voltage of an SEM, or 200 kilovolts, a typical acceleration voltage of a TM. And here we have the wavelengths of these electrons that range from 2.5 picometers to 12 picometers. And if you compare this to photons, for example, X-rays or visible light here, we are at 100,000 picometers. So because we have such small wavelengths, we can image smaller and smaller materials. So here you see an example of a 9 nanometer gold nanoparticle imaged in a transmission electron microscope, where the typical wavelength is somewhere around 2 to 50 picometers. So we can really image atomic resolutions. As you see, all these little dots here, simply speaking, are atomic columns. So now we know that we can utilize this short wavelengths. We also need to understand what type of interactions we have when electrons come into contact with our minerals or with any matter in this sense. So for this, we can use the Bohr model of an atom where we have a nucleus here. We have different shells where electrons are flying around. We can name these shells, typically called K, L, and M shells. And now we bring in electrons to interact with these atoms. One thing that can happen is that an incoming electron here in red hits an electron on one of the shells and kicks it out from the shell. And this we call a secondary electron, which will give us topographical information. As I mentioned earlier, one of the basic informations we can get within a scanning electron microscope. But also, when an electron relaxes from a higher shell onto this shell, we can get X-rays emitted, and there will be a dedicated lecture, as I said, on using X-rays for chemical analysis later today. Then, if an electron comes in and interacts with the nucleus and is backscattered and comes out of the material again, we call this backscattered electrons, and this gives us element contrast. So, to further understand it, we have to look at a diagram such as this one here, where we have the sample and the incoming electron beam, and let's have a look where different interactions occur. So secondary electrons, I already told you, are those that are emitted and kicked out of a shell when an incoming electron, and this comes from the top of the sample, and the size of this area here depends on the acceleration voltage, but in general it gives us topographical information. Backscattered electrons come from much deeper.
Is working now.
You please.
But the first is on SEM, so maybe not so.
Could you please mute your microphone so we don't have interference? Thank you. So where was I? Okay, backscattered electrons come from much deeper within the sample and give you atomic number and phase difference contrast. And then again, of course, X-rays are emitted from deeper within the sample, and we will talk about this in a later lecture. So here are two images of the same material, of the same area, but one is collecting secondary electrons on the left-hand side and backscattered electrons on the right-hand side. And you see the immediate difference. We have a material with some particles on top of it. In the secondary electron mode, we see topography. We see actually that these particles here are topographically higher on a material that actually has quite a bit of topography itself. But in the backscattered electron mode, when we collect those electrons that interacted with the nucleus, we actually see that this material here is made up out of three phases. The substrate at the bottom has an intermediate grayscale level. Then we have a very dark substrate. These little particles emerge into a very bright phase. So what we can already say from this image is that we have three phases with three distinctive atomic weights, and that the bright phase is the heaviest atomic numberly speaking. So it could be an iron oxide, whereas these particles could be carbon based on a quartz crystal. So you get the contrast in the backscattered electron image scales with atomic number. So as I said, you can do much more, and as we have only limited amount of time in this lecture, later after my lectures, we will specifically talk about X-rays in the scanning electron microscope and how we can use this for chemical analysis. This will be a lecture today at 10:15, followed by a lecture on how we can use crystallographic information with something called electron backscattered diffraction inside an SEM at 10:45. So now that we understand scanning electron microscopy in a nutshell, we can not only use electrons to image materials, but we can also use ions. So for example, gallium ions in this case, to image and manipulate materials, something called focused ion beam scanning electron microscopy, which is really handy for a lot of more advanced characterizations of Earth materials and also for sample preparation. And I would briefly like to discuss how we use such a focused ion beam scanning electron microscope that you see here on the right hand side in our lab in Utrecht. But what this is now, instead of only having one source, an electron gun, you also have a gallium ion source, and you can tilt the sample between the different sources to do very interesting techniques, such as three-dimensional nanotomography or the preparation of really thin samples, extremely site-specific, for later high-resolution analysis. So let's have a look at what happens when not electrons hit the sample, but when ions, and in this specific case gallium ions, hit a sample or a substrate, and what happens. So very similar drawing as I've shown you just a minute ago. Here now we have an ion beam hitting the sample, and we have numerous interactions. We can also have secondary electrons that emerge from this ion matter interaction that should also show you topography. But we can sputter away atoms and ions, so we can cut into the sample, which I will discuss in a minute more. But we also have implantation of gallium, which is these three little spots here, which you should keep in mind that you actually now implanting a different ion into your material, which may have repercussions later on if you're specifically interested in gallium, for example. And because of this implantation, we also can get amorphization and recrystallization. So we can actually disturb the sample. But in general, we talk about three different modes: imaging, milling, and deposition. And all of this can occur simultaneously. So the FIP is like a little nano scalpel. You can actually mill into the material with extreme resolution. This is shown here. We can actually scan the ion beam across the sample and cut into the sample with extreme precision. And of course, the resolution of this cutting is smaller at lower currents, but it takes also much longer. So we can not only image and cut into the material, we can also deposit other types of material. So if we have what's called a gas injection system, we can actually inject gases, and typically this is platinum, carbon, or tungsten gases into the material, and with the ion beam we can steer the material to deposit specific layers. So people have gone crazy with this. For example, I found this picture quite a while ago on the internet. This is a tiny toilet made probably out of platinum deposited inside the gallium with the gallium source and the gas injection system inside the focused ion beam. Or for Star Trek fans, this is Starship Enterprise deposited using a FIP SEM. But here on the right hand side, you can also see the precision of cutting into material. This here is a little device. This is about 100 micrometers here of cut into a material using the FIP. And I should also say that this toilet here is about 200 micrometers inside, in size. So you have a very powerful technique, not only using the electron beams to image materials in the using secondary electrons to get topography, or backscattered electrons to get element contrast, or using X-rays. You can also manipulate material, cut into the material, or deposit material on top of your mineral. So why would you do this? Before I answer that question, a quick look into the chamber of such a focused ion beam scanning electron microscope. This is what you see here. This is a view of one of our older FIPs. Here we have the electron source. Here you have the gallium ion source. You see they are at a specific angle to each other. Here we have the gas injection system, and also a nano manipulator needle, which I will come to later, that allows you to actually extract tiny samples from your larger samples, which could be a thin section of a rock, which probably most of us are dealing with. So one thing you can do with the focused ion beam scanning electron microscope is to do simple cross-sectioning. Here on the left-hand side, you see an image of, this here is an experimental sample. It's calcite, so calcium carbonate, that somebody has deformed inside a laboratory. So there don't need to be always natural samples. This is an experimental sample, and it produced these very flat surfaces. And the person wanted to understand what's actually happening below the surface going into the sample. And for this, we cut a trench into the front using the focused ion beam scanning electron microscope, and then actually had a look from the side. So you not only now can image the surface here, but you can also see what's happening below the surface. So you get a better three-dimensional impression. And you see here, typically these trenches are on the order of several tens of micrometers. The larger you make them, the longer it takes with a standard gallium ion beam source. Just a small side note, nowadays you cannot only have gallium ion beams as a FIP, but you can also have plasma FIPs, which allow you to ablate much larger areas on a millimeter scale. But nevertheless, once you have cut into the sample, you can actually use the power of the electron microscope to get very high resolution images. Here the field of view is 5 micrometers to try to understand what was happening below the surface. So this is conventional cross-sectioning within the focused ion beam, making the full power of manipulation using the gallium ion source in the imaging of the electron microscope. But you can not only do one cut, you can also do a sequential slicing of your sample. And you must imagine this similar to a salami, where you start slicing the salami up, but every salami slice is maybe only of the order of 5 to 10 nanometers in thickness. And then we have the ion beam cutting the sample and the electron beam imaging each slice sequentially. So here on the right-hand side, you will see a little video of how this works, looking down the ion beam column. So we're looking down this line here onto the sample. This here is a platinum layer that was deposited onto the sample to protect the area of interest. This now probably also becomes much clearer why we need a gas injection system. We have to screen our sites of interest from the implantation of gallium or the general abrasion of the gallium ion beam by depositing a layer on top of it, which then keeps that area that we want to image undisturbed. And then we have this little LY here, for example, is a little fiducial marker that allows us to correct for image shifts and keep the beam quite steady. So now if we play this video, you see how we have a sequential cutting throughout the material, and then the electron beam is imaging each of these slices. You also notice that that this technique is destructive. So once we're done with this slice and view or this sequential slicing, the material of interest is gone. So we need to make sure that actually, if you have only very little material, that this will work. But most of us that are working, for example, with rocks, we probably have enough area of interest where we can do this. So the disadvantage is, compared to X-ray tomography, which we will learn about tomorrow much more, is that FIB tomography is destructive, but it allows us to image at very high resolutions with the electron beam down to nanometer scale. So what you see here is a video of a feldspar that has porosity inside. You see all these blacks here is porosity. This is actually a backscattered electron image. You see there are at least two types of phases here, the darker contrast one and the lighter contrast one. This is about 15 by 15 micrometers. And now you see this sequential slicing and imaging throughout the material, very similar to X-ray tomography if you have looked at that before. And now we can reconstruct this pore space or any area of interest in 3 dimensions to analyze this at very high resolution. So from this, for example, you can get the size of the pores. This here is the pore channel diameter against the probability density to show that in this case, for example, most porosity is about and below 100 nanometers. But it's just one example of many things you can do with this type of 3-dimensional FIB SEM tomography. But in general, you can go from conventional 2-dimensional SEM imaging, such as this backscattered electron image here that shows you the atomic number contrast, to 3-dimensional FIB SEM nanotomography, eventually to physical properties using numerical modeling. And we will learn more about how you go from images to 3 dimensions and visualization and data treatment tomorrow, because the techniques that we use here to visualize these 3-dimensional volumes are the same as in X-ray tomography. So there's actually a lecture tomorrow on image segmentation or post-processing of X-ray CT data, which will also be applicable for the data that comes from a focused ion beam scanning electron microscope. Right, now that we know that we can manipulate materials using the focused ion beam, we can not only do tomography, but we can actually prepare samples, very site-specific, to go to a transmission electron microscope. Because, as you probably noticed in the first picture, in a TEM, we have to go through the sample. And this is only possible when samples are really thin. So typically around 100 nanometers to become electron transparent. And we need to make these samples. There's various methods, but the state-of-the-art method also that allows us to be extremely specific. I want to have my sample right here across an interface and not 5 micrometers to the right, is in the focused ion beam scanning electron microscope. So what you will see here is a little video again of how such a preparation works. We have a sample. We will cut a trench using the focused ion beam in front, but after we deposited a platinum layer or a tungsten layer on top of the sample to protect the area of interest, now we're building a trench in the front. You already see here some porosity, for example. Now we cut a trench in the back. We bring in the gas injection system again, and we bring in a needle, a nano manipulator that we can control with our mouse and keyboard. And you will see what we do with this in a second. This needle now is brought to the sample, and we have cut out this sample at the bottom on the side and also here a little bit, but we leave a little bit of area left over, so this is stabilized. And now we will approach this needle onto the sample and use the gas injection system to weld the needle to our sample. And then we cut out this little bit on the right-hand side. right here. And now we have a sample that we can lift away from our mineral, for example. That is on the order of 15 micrometers by 5 micrometers in this case, but it's still a micron or two thick. We will bring this to yet another holder that we can pick up with tweezers, and we will put the sample onto this holder, welding it again with our platinum gas. Then we cut off the needle. And now we have this tiny sample across an interface, through porosity, across a grain boundary. There's infinitive amount of options. We will take away the gas injection system and the needle. And now again, we use the gallium ion beam to make this sample extremely thin, typically on the order of 100 nanometers. So you see the sample is still relatively thick, 1 to 2 micrometers. And now we will thin it down until it is very thin, 100 nanometers about. Then it becomes electron transparent, and we can now take this sample with this holder and transfer it to a transmission electron microscope. So here you see one of these samples in a TEM already, and I will talk about TEM more in a minute. But now the sample is electron transparent. Actually, here on the bottom is the platinum that you see, and a little bit of the needle that has not fully come off is seen here on the left-hand side. But now we have, with focused ion beam electron microscopy, we also have a very powerful technique to extract extremely site-specific samples from anywhere, really, to take it to a transmission electron microscope to observe nanoscale and atomic phenomena. So that brings me to transmission electron microscopy. We've seen one state-of-the-art technique of how we make really thin samples from rocks and minerals. Of course, if you deal with, for example, with nanoparticles already, you don't have to go to this elaborate focused ion beam scanning. Most nanoparticles are already electron transparent by itself. But now let's put such a foil into the transmission electron microscope and take electrons through it and have a look what happens when electrons go through a material. So to remind you, here we have the two very basic interactions within a scanning electron microscope. These also occur inside a transmission electron microscope, but we're much more interested in those electrons that are transmitted. So we have a beam that goes straight through the sample and is not interacting with our material at all, or we have electrons that go through our sample, interact with the nucleus, and are elastically scattered. That means they change their path. So here we see this representative in this little kink. With elastically scattered electrons, we can do different types of imaging. We will, in later lectures, in more advanced courses, talk about specific bright field and dark field imaging. We can use it to do diffraction as well. And diffraction in a TEM will be a specific lecture later today. We can also use those electrons that actually go through the sample. They don't change their path, but they lose energy. So these are called inelastically scattered electrons. And this is a very powerful technique for qualitative and quantitative element analysis via something called electron energy loss spectroscopy. We not only can get chemical informations, but also nearest neighbor information. There's an infinitive amount of possibilities using electron energy loss spectroscopy to go very deep into material behavior. And of course, we have secondary electrons and X-rays transmitted, and I will very briefly talk about X-ray analysis in a TEM in a minute. What we need to know is that there are two basic TEM operations. First of all, in a TEM, our electrons are coming in parallel onto our sample, in this case here called specimen. And we can form an image on our screen. And here on the right-hand side, you see, for example, a bright field image of a feldspar that shows twins. You can see this here. So the different twins diffract differently. The paths have changed of the electrons. You get this diffraction contrast, and you have an alteration zone here in the middle. This is in the imaging part. But if we take the back focal plane behind the objective lens, we can also use this to get diffraction information. So always images and diffraction are happening at the same time in a TEM. It depends on what your TEM conditions are. Either you are in imaging mode or you're in diffraction mode. So you view the real image or you view the back focal plane of your material. And we will talk more about diffraction in a later lecture, as I said, but typically this is how these type of diffraction patterns look like. Some of you may have already seen this. This is a single crystal diffraction pattern here of, in this case, olivine, and the distances between these dots tell us something about lattice constants within our material. We can also look at polycrystals, then we get ring patterns here. And we can look at orientations, for example, common orientations of many particles together, or simply analyze what these particles are using diffraction. Or we can get amorphous diffuse patterns, as you see in this case here, to say something about the degree of amorphization, or simply is there amorphous material inside our sample. Lastly, we can go to very high resolutions, and I won't go into detail here in this lecture, but we can go, and you've seen already one picture of these gold atoms. We can go to high resolution phase contrast imaging, where we can now look at materials such as this one here. This is a chrysotile fiber. This is a serpentine. It actually has a tiny gap in the middle here, and you can study the atomic configurations within this mineral, and the distance, for example, between these layers here tells you something about the real distance of your crystallographic spacing. Quickly to electron energy loss spectroscopy. So not only using the elastically scattered electrons, but also these inelastically scattered electrons. In earth sciences, for example, is used a lot to determine the oxidation state at very high resolutions of, for example, here pyroxene, and you see a spectrum of the energy loss against the intensity. We're looking at, in this case, iron, and you see here how the spectrum changes from a dominant peak here to a more dominant peak on the other side. And this tells us something, and is can be quantitatively expressed about how much iron 2+ to iron 3+ is in our pyroxene. So we have a very powerful technique to, for example, look at oxidation states at very high resolution. So in a TEM, we not only have transmission mode, so the electrons are coming in parallel onto the sample, we can also converge the electrons and do something called scanning transmission electron microscopy. So similar to an SEM, now we have a beam with a point here where we converge these electrons and we scan them across this really thin sample, and we collect the information that comes out at the bottom below the specimen. Scanning electron microscopy is quite powerful in the sense that it can provide us with additional information besides diffraction contrast, or besides diffraction contrast or diffraction patterns. You can also get diffraction patterns using convergent beams. But the main thing I would like to highlight is you can get information of those electrons that are very highly scattered below the sample, something called high angle annular dark field. And simply speaking here, this is similar to backscattered electron imaging inside an SEM, where these scattered electrons provide us elemental Z contrast information. So here on the top right-hand image, you see a quite high resolution TEM image with probably a field of view of about 5 micrometers. And we can, the contrast here scales with atomic numbers. So we know already we have 3 different phases. Again, we have this very bright phase here, which could be an iron oxide. We could do an X-ray analysis at a later point in a fairly homogeneous matrix. And then we have this intermediate phase here that you can see, which might be, in this case, is actually clay. But we have a similar technique inside an TEM, where we can look at element contrast images to an SEM. And we can actually do this at very high resolutions. Won't go into much detail again here, but you can do a high angle annular dark field imaging at atomic resolution. And this is quite an impressive image here of phase boundary between two materials. You see all these little dots here are simply speaking atom columns. And you see that actually the contrast of these columns varies, which is the direct representation of different elements within this material. So now that we can scan across our sample, we can also produce element maps using X-rays. And again, we will talk about this in the next lecture, but you can do element mapping at very high resolutions inside an SEM. In this case, for example, it was a study on deformed quartz clay mixtures and seeing where the clay wraps around the quartz. You can map this out very nicely in a TEM on a focused ion beam sample. And again, you can not only do this at a high resolution of several microns, you can actually take this to atomic resolution and do atomic energy dispersive X-ray mapping using a scanning transmission electron microscope. This is a recent example here. You see the resolution now. We have scale bus of 1 nanometer, and they map out individual atom columns using EDX. And this is something we will look at in later workshops of how this could be done. But again, I wanted to show you that you can take EDX and an element contrast mapping all the way down to atomic resolutions. So this leads me to the end of a very brief introduction into scanning and transmission electron microscopy. If you would like to know more about this, we will provide all these lectures as PowerPoint presentations on a secure server that you will get access to later. We will also upload these lectures onto YouTube so you can watch them again. But if you want to do some more reading and you need some more information, then go to our webpage on www.excite-network.eu. You go to Access and you go to the microscopy knowledge base, which is an ever-expanding dictionary of helpful web pages around electron and X-ray microscopy, where you can find a lot of information. A little commercial and a great web page is also from our Australian colleagues at Microscope. So if you go to microscope.training, you will find a lot of information about specifically electron microscopy. How does a scanning electron microscope work in detail? You can follow online lectures and trainings here. Fantastic resource to use. And also, if you have questions, we can discuss them now. And with this, I would like to thank you for your attention.